So our first speaker today is Molly Cummings. Molly Cummings got her her undergraduate degree at Stanford. And she was telling me she did her PhD at UC Santa Barbara, and she worked with John Endler, where she learned a lot about guppy coloration. Then she went on to do a postdoc at UT Austin, where and then she stayed there as a faculty member and has been there for years. She studies coloration in lots of different animals, from fish to frogs to insects, and she studies coloration in lots of different ways. So she's interested in why animals are colorful, how they stand out from the crowd to attract mates and avoid getting eaten. She also studies how animals blend in cryptically to avoid getting eaten. And one of my favorite things that I found on her website is she has some really cool work on how fish have these special cells on their skin that help them reflect polarized light in new ways so they can really blend in the ocean. And one of the reasons I thought this work was cool is, not only that's amazing, the way that fish camouflage themselves, but it actually has exciting practical implications. Apparently the Navy is interested in using these techniques to help submarines blend in. So another great example of work you wouldn't necessarily think being practical turning out to be amazing. So we're really looking forward to her talk today.
Thank you, Liz, for that beautiful and very kind introduction. And also thank you to the entire committee, organizing committee, for inviting me here. I'm hugely honored, and I do think, as in the nice introduction of the symposium, it's such a great forum and so important. I hope that more universities follow your lead in taking this on. So I'm very excited to be one of your keynote speakers for the Ecology and Evolution of Color Symposium. It is those two kind of tactics or strategies or lenses on color I've approached from a number of different angles, so I'm very excited to be here. And what I've decided to do is that because my particular path through science has been associated with a couple of pretty famous scientists who are game changers in the field of signal evolution, I decided to give some historical context first from this whole field, and then move into what we're currently doing, and then move into the future, which is really you. And I really want to point out that game changers, the kind of game changers I'm going to be talking about today, changed the field in the 90s, but you in this audience are going to be changing the field now 30 plus years later. So it's kind of really neat to be here. So I will be giving a pretty quick overview on a lot of great studies that have approached this question, including highlighting the next speaker, Natasha Bloch, with her beautiful Warbler work. But I'll be doing a lot with fish. I'm a fish head, and you're going to see why fish are feature prominently in this piece of this body of work. And I'm also going to even go into amphibians as well. So to start off, I'd like to give you kind of the origins of this idea of sensory drive and what it is that I'm talking about. So, and then I'll go into some specific examples, and then I'll go into, the future, or what I see as the future of this field. So in terms of the origins, can you still hear me when I walk over in this direction? In my mind, it really all began, at least from a very comprehensive theoretical framework with John Endler. In the early 1990s, he put forth, this is one of a couple papers that he really laid the foundation for this concept of sensory drive. And it's pretty comprehensive in that it goes from the environment to the sensory systems to signal evolution as well as to behavior. And a very important feature of this is natural selection, that we can look to natural selection to be driving sensory and signal evolution in a very predictable direction. And I want to point out why this was a game changer at that time. So at that time, if you think of evolution of mating signals through female choice, the models were somewhat arbitrary in terms of the specific features that would evolve for males to attract female attention. But with this new paradigm, sensory drive, it was no longer arbitrary. You could actually start predicting the direction of traits that were used for communication, and that's why it was a game changer. It might seem perfectly intuitive at this point and make complete sense, but at the time, it truly was a game changer in sexual selection. And this is John Endler's original model. It was a nice, simple to find or follow flow chart. And it all really began with the environment. So the environment and particular conditions of the environment, usually biophysical conditions, would set the stage and place selective constraints on sensory systems. So sensory systems would evolve attributes and receptors to detect certain features that would stand out best against their particular environment, usually in the context of natural selection, to find food or to avoid being eaten. And then, because of this selective constraint on the environment, on sensory systems, biases would evolve, and those biases in the sensory system should be able to predict the evolution of signaling traits that would emerge amongst the signaling sex. So essentially, it was this coevolution of sensory systems and signals with the environment that really underlie sensory drive. And I think we might as well go to John Endler himself in this great sentence where he sums up and introduces the concept. These suites of traits should co-evolve in predictable directions determined by environmental biophysics, neurobiology, and the genetics of the suites of traits, hence the term sensory drive. And I'd like to point out that simultaneous to this time period of John Endler's sensory drive was Mike Ryan and others of sensory exploitation model. So you'll notice the areas that are highlighted or outlined in bold are portions of the sensory drive model that actually encapsulate sensory exploitation. And sensory exploitation is a very similar idea. It starts from the point that females or the receivers in a communication dyad already have these biases, usually through natural selection, but it didn't really get into the details. Female preferences then that are deriving from kind of the neurobiological sense evolve outside the mating context, usually through natural selection, and this then places selective constraints on what type of signals should evolve. And the emphasis in the sensory drive model is the historical component. So between these two models, we have ecology and evolution of color or signals. Okay. So real quickly, I want to now run through, or walk through, hopefully stroll through, a few, I think, pretty comprehensive examples of the sensory drive model that all focus on color. Before I get there, I want to bring up two different reviews that I've been a part of to look at the evidence for this model. And a few years ago, Mike Ryan and I decided to look at the evidence for sensory drive and perceptual biases involved in mate choice. And so we took John Endler's original model and kind of added a few bits, but then went through the literature to find evidence at different points or steps in the model. And so each number you see on these various flow arrows represent a different study that provided evidence for the different steps in the model. And the different colors of those numbers depict which sensory modality this particular study was playing in. And for today's purposes, you might be happy to see that a great number of these studies were red. So we ended up finding quite a bit of a bias towards the visual modality. And we did this just kind of, we didn't statistically examine this, but that's what we found at the time. More recently, John Endler and I decided to actually write our first co-authored paper together. It only took us, I'm not going to admit, I'm the oldie of this group today. So I've been working with John since the late 1990s. And this is our first paper that we've co-authored, which is pretty neat. So anyways, we took his original model, we shaped it up a little bit, and we decided to split it into two of its biggest components. The sensory components, which we highlighted in green right here, starting with sensory components that are influenced by environmental conditions, and how that shapes mate choice and other kind of perceptual biases, and they were driven by usually detectability of food. And then we looked at signal components, which effectively make up the latter or the bottom portion of this flowchart. And we looked for studies that evaluated, this is a big concept model. It's really difficult to test all features of it. And yet, so people have gone out and tried to usually look for evidence in certain portions of this model. And so we evaluated studies that just looked at the sensory components to see if it provided evidence, just looked at the signal components, whether that provided evidence, and then studies that tried to attempt to do a more full, comprehensive, both suites and how they co-evolve with the environment. And so we examined this to determine whether or not there was any taxonomic bias, whether or not there was a habitat bias, or whether or not there was a modality bias. And when we looked for taxon, we kind of grouped them into their large classes, and we looked across 156 studies. I am sure we missed many. We really tried to focus on ones that had a strong environmental component involved. And due to our own kind of vertebrate biases, I'm sure we missed a lot of invert, but we tried. So how you look at this, this histogram is the clear bars show support for the sensory drive model. The black bars, no support. The top set of figures show the sensory component studies, and here are the different classes down below, then the signal components, and then both. And what you may notice is that, well, first of all, birds actually were showed the greatest support in the signaling components. They are the top taxon that have been examined showing support for this particular model. But you also might notice that fish feature prominently across all three of these ways to examine the model. They are the top taxon in the sensory components, strongly represented in the signals, and also, once again, strong in the full complete model. So today I will let you know, no spoiler alert here, that I will be focusing a lot on fish. Yeah. So we then looked at whether or not there was evidence by modality. And I apologize to anyone looking at olfaction or vibratory cues or tactile cues or magnetic reception, but we just focused on the two big modalities that have actually been examined over the last several decades. We are now getting incredibly good at looking at olfactory cues, and I think some exciting work is coming out in that direction, but we didn't include it in this particular review. And what you can see here, there's a couple interesting points here. Well, one is that the vision dominated all three, but you also see in the no support column a fair amount of non-support in the auditory realm, and I'm going to get to that in a moment why that might be. But today, again, and because we're interested in color, no surprise, I'm going to be focusing on the vision portion. So when we looked at evidence by habitat, what we found, which was particularly interesting, is that actually terrestrial habitats show some of the greatest support when you just focus on the signal components, which is really interesting to me. But it's also statistically significant that they show the greatest non-support as well. So there's a lot of auditory, particularly in the bird and amphibian and invertebrate world, where there's no connection with environmental biophysics. And the idea there is that the social environment itself is creating its own environment, and that's driving what really matters in those particular signal components. But today, because I'm focusing on fish and vision, I'll be doing a lot in the aquatic realm. Okay, so let's get to the aquatic realm. And why is there a whole lot of support in aquatic versus some of the other terrestrial environments? When it comes to vision, it's quite a significant environment that drives, in a very important way, constraints on signal and sensory systems for a number of reasons. One is the water molecule itself differentially absorbs wavelengths according to light according to wavelengths. So as you go deeper, you lose both the extreme short end as well as the longer wavelengths of light. And in the worlds that I work with, my PhD work, I actually work with surf perch in the kelp forest of California. That is one of the most extremely variable optical environments because not only do you have a depth component, you've got a canopy component of an underwater forest, and on top of that, you have variable phytoplankton communities that pop up with different compositions. So you can have red blooms, green blooms, and some combination. So on a given day, your world can look like this at the same depth, habitat, and another day it looks like that, and another day you have an image that would be terrible, wouldn't even make a good photo because you couldn't see past your hand. Meanwhile, when you look at a terrestrial forest environment, there are changes. In fact, the reason why I started working with John Endler, there's a number of reasons. I read his Guppy work, and then I read his Light in the Forest paper, and I got all excited because I had been working at Hopkins Marine Station where there's this kelp forest. I typed him a letter, this is how old I am, asking him would he be interested in taking on a graduate student to test sensory drive in the kelp forest. He was pretty excited. But what's interesting is the light, there are differences, but they're more subtle in the terrestrial environment. And we're going to see why some beautiful work by Natasha has shown that it does make a difference, these different light environments. But to really find that, you've got to take a broad phylogenetic approach because you need strength in numbers, because the optical environment is a little less severe than underwater. Okay, so I'm not going to go through all of these, but here's just a nice look at a whole bunch of fish where it's been examined and found support for the sensory drive hypothesis. And here I want to point out that people have looked at this in different ways. As I mentioned before, that's quite a comprehensive model, a lot of different components to it. It's pretty impossible to be one group of scientists able to test every bit of it. So some have kind of left out the actual biophysical environment and focused on food. These carassins do amazing things with pectoral fin ornaments that are shaped like ants, and that's what these fish eat. The cichlids, which I will cover a little bit today, have done a great job of examining kind of real comprehensive approach, and other people have tackled different elements of this model. So not surprisingly, I'm going to first start with the case study that I worked on with the surf perch. This is work I did for my PhD, and I went out to the kelp forest and would measure light for a couple of years. It was a pretty awesome PhD to go scuba diving on a regular basis. And I measured the optics of their different species of surf perch, where they occurred in this kelp forest. So I would take light measurements and just point out which species was present. So it ends up that the different species have different optical environments that vary both in brightness components as well as color components. And then I collected these retinas, went over to England to work with Julian Partridge, and we measured the visual sensitivity of these different the visual pigments in each of the species. What ended up being a wonderful thing for me, and obviously evolution thought it was a good idea, these fish were dichromats. They only had 2 cone classes. It's great speaking to a group on color because you know what that means when I talk about cone classes. They had a shortwave sensitive cone class and a longwave sensitive cone class. And there was huge variation across the lambda max, the peak sensitivity of these visual pigments. So much so, if you just look at their peak sensitivity in their shortwave sensitive cone, the peaks between the species varied by 30 nanometers, 30 to 35 nanometers. Now, all of us in this room are probably familiar with our own human visual pigment sensitivity spectra, and you may recall that the difference if you're blessed enough to have a middle and a long wave sensitive cone class in your retina, which hopefully 90% of you have, the difference in peak sensitivity is about 30 to 35 nanometers. So here, between species, the difference in a single class of photoreceptors varied dramatically. And so if I took a simple approach and just looked at the lambda max of their visual pigment sensitivity of one cone class, this works for both, and then compared it took all those hundreds of measurements across their habitat and just calculated a maximum flux, average maximum flux, there was a simple correlation. So something about the environment was driving differences in peak sensitivity. I then took what I would consider a slightly more sophisticated approach, and I evaluated their sensory bias through a visual model that used dichromatic model for both brightness and color. And fortunately for me, someone actually, a friend of mine, Giacomo Bernardi, had calculated the phylogeny of the surf perch. I was so grateful because then I could estimate the peak sensitivities of the 2 photoreceptor cone classes of the most common or most recent ancestor. And then also a whole bunch of marine ecologists had figured out what these fish eat. I didn't want to do all that gut contents, but someone else did. I was very appreciative. and they all search for the same red algae. That's where they find their food. So it was beautiful. I had different environments. I had the same common food source, and I could estimate the ancestor. So it was very fun and satisfying to then evaluate a sensory bias based on the divergence from the ancestor to find that same food target in their now diverse physical environment. And what was neat, and this is the neat part about a dichromatic system, is that if you move one way versus the other, you're either going to get a gain in brightness detection with a loss in color detection, or you're going to get increased color detection with a concomitant loss in brightness detection. And it's simply whether or not you overlap your cone photoreceptor absorption peaks that would increase your brightness contrast, or if you bring it further apart. So that created this nice dichotomous direction from their ancestor. And two of the species went in a direction that increased their color contrast detection relative to the ancestor in their environment. And three of them went more for brightness. And I'm not going to show this here, but some of that had to do, or I would hypothesize, has to do with how much brightness variability I detected in each of these color-biased species. So the next thing I did was then evaluate their signals relative to, you can do it either to the, well, to an estimated ancestor or an average between their peak reflectances. So here are two species, here are the species down below, that share blue as a featured color in their nuptial coloration. But you can see they've diverged in their peak reflectance of their blue in quite a dramatic fashion. And so I can evaluate the detectability of their species level reflectance, as well as model what would be the best reflectance property to be seen in brightness contrast channel or color contrast channel. And so I can evaluate the signal bias. And what was pretty beautiful or satisfying about this is that the two species who had a color contrast visual system biased for color also had reflectance properties that were biased to be detected by color in their environment. And conversely, the species who actually had a visual system biased for brightness detectability had signals that actually showed a bias in brightness detection. So it was a very satisfying full circle study with the surf perch. One of the most satisfying pieces of evidence for the sensory drive model has come from the cichlid world. And this is work with Seehausen and a number of other people, Martina Mann and others, where in this group of fish, they've looked at color evolution and sensory evolution across 5 different islands and a number of different species. And they found a depth-mediated light gradient, light male color gradient. So we've got distance from shore, orange ratio as they go kind of deeper down. Their distance from shore is a depth measurement in this particular system. And as you get deeper, you lose different portions of the spectrum, as I was talking about before. And what they found is male color and female preference were linked across this gradient, and they identified the functional genetic components that actually were mediating the sensory drive speciation in sympetry. These guys only vary in about 5 to 10 meters from each other in terms of their habitat use. So it was a very satisfying study. And more recently, Martina Mann has done some transplant experiments that reveal differential survivalship across these different morphs in these different depths. So Martina, who actually did she's not Martina, did a postdoc with me on the poison dart frog work, which I'm going to get to in a little bit. But she's recently done this beautiful work where she simulated the different, these are the simulations of the different morphs in either shallow or deep light environments. And then she did this really beautiful survivorship experiment determining which morphs survive greater in the different environments. And it was really completing the circle why you would have phenotypes varying by depth. Another classic system for sensory drive is the Stickleback work. And Jenny Bowman put this beautifully on the map following earlier work by others. And she looked at 6 different populations, and she found that both the visual sensitivity and the male nuptial coloration and female preference co-varied across these different populations. And more recently, some work from a graduate student that I co-advised with Dan Bolnick, he took this and expanded it. He looked across 15 populations. He looked both within a population as well as between populations to look at that depth by male gradient. And he also finished it out with a transplant experiment. Let me show you his work. So as I mentioned, he did both between and within populations. And he identified within depth gradients between male, color, and the light operations or the light conditions. And what was neat, and this differs from the cichlid work, it all goes in one direction. Stickleback work, you actually have populations where the optics go in the opposite way. So what do I mean by that? There are some environments that favor blue in the shallows and red in the deeper zone. When I say deep, we're talking across a 2-meter gradient. So this is really exciting to find this kind of micro scale variation in color with depth. And because he looked at 15 populations with different compositions of tannins or other kind of organic and inorganic matter in the water, you change the optics. So it was this beautiful natural experiment to see, okay, is it depth that's driving this, or is it the specific optics at depth that is driving this? So for instance, we've got deep males here that are a little more blue biased, and shallow males in this population that are more red biased. And then in another lake, it's the opposite, where the shallow males are blue biased and the deep males are orange or red biased. He then, within one of these environments, he actually then did a transplant experiment to see which direction, males who were actually stuck in one depth zone, would they change their colors? And they did. And they changed them in a unique or a specific direction. If males were found in the deep, they became more conspicuous with chromatic contrast. If they were stuck in the shallows, they actually minimized their color contrast, which is interesting and is somewhat consistent with what the cichlid world is showing. Okay, so even though I'm a fish head, I'm going to do my part to kind of come out on land and look at some wonderful terrestrial examples. And as I mentioned before, one of the reasons why I even got into this was because of John Endler's wonderful paper, Light in the Forest, the color of light in forests and its implications in 1993. And this is where John evaluated specific microhabitats in terrestrial forests and quantified the light conditions in the spectrum of light and noticed that it would shift the relative composition of the ambient downwelling light. And about the same time, Karen Marchetti was doing some seminal work with the warblers. And she noticed, just looking, she didn't take any fancy measurements of the light environment, but she looked at signals of males. And what she noticed is that the amount of the white bar on various warblers varied based on the kind of habitat they had. Oh, sorry, she did take, it wasn't fancy, but light flux measurements in these different habitats. And what she found is a negative relationship between the number of bright patches on the males and how bright their environment was. So in darker environments, males brighten up their signals, and vice versa, in brighter environments, they darken their signals. So this kind of led the stage for current game changers like Natasha, who's going to be speaking next, who started to examine whether or not there were any opsin shifts across habitats. And what she found, at least in females, not so much in males, is that this is just a long-wave sensitive cone. She found differential expression based on where those females were found, whether they were in coniferous forests versus shrubs, was a strong predictor of how much of that long-wave sensitive opsin they were expressing. She then went even more broadly with her comparison. It's really a phenomenal, comprehensive piece of work where she looked across old world warblers, finches, and habitat use and looked at the shortwave sensitive cone and found these big, across that broad phylogenetic comparison, big changes in the shortwave sensitive cone expression based on whether or not these animals were in understory environments, arboreal habitats, or open environments. I'm sure she's going to expand on that some more. So the next thing I'd like to do is really focus, and Elizabeth mentioned this in the beginning, that I do also how I look at how predators influence the shape of color. I'd like to look at this little corner of the sensory drive flow chart, and that's examining how predators influence signal evolution. I'm going to start with the terrestrial side of things with my poison dart frog work. I started working with poison dart frogs, actually first a group down in the Amazon, but then moved to these wonderful, this wonderful group, Pumilio, Ufaga. They are the strawberry poison frogs, and they are actually, Ufaga Pumilio, they're actually found in this archipelago that's not actually popping up very well on this slide. But in this, most of the mainland across Nicaragua, Costa Rica, and Panama has a single morph, and it's a strawberry poison frog, or blue jeans frog, so has a red back and blue legs. And I'm smiling because J.P. Lawrence is in this room, and he actually came out to work with me on this project many years ago, so it's very satisfying to see him in the audience. But in this little group of islands, this archipelago and off of Panama called the Bocas del Toro archipelago, the different islands have evolved different morphs. And remember, these animals are using their colors to communicate to predators that they're toxic, please don't eat me. So it's an aposematic warning coloration all the way back to both Bates and Mueller. The idea of diversifying your warning coloration made no sense, right? You want to actually, let's all group together and have the same signal, and you dilute having to teach your predators. But here, we had an aposematic system that had huge variation. It didn't make a lot of sense. And so we've examined this from a number of different point of views, both natural selection and sexual selection. And from the natural selection side, we asked, okay, well, maybe these different islands have different types of predators. How do different potential predators see these colors? And so we evaluated using visual modeling techniques what the different morphs would look like from a bird's point of view, a crab's point of view, and a snake's point of view. And we also then wanted to evaluate, tried to guess. We don't do the amazing work. We did the sensory ecologist's cheap way of approaching this problem. I'm going to get to our solution of this problem in a moment, which is simply looking at visual models. I want to do a shout out to Allison's beautiful work and her system, which I learned about, I sheepishly have to say, last night, but it was amazing. And she does some really hard work where you actually use models and test it directly with the predator. We did it a little less labor intensively. We actually used visual modeling techniques to evaluate who is the best predator who can actually see the relationship between their conspicuousness and toxicity the best, okay? So let me show you that data in a moment, but I also want to point out that it's a coevolution between signals that were presumably first evolved to communicate with the predator. Also, the females are paying attention to this, and the females appear to be driving the variation as well in terms of they really care about the brightness across at least 4 different populations. Okay, so how did we do the cheap way of using sensory modeling to figure out who the predator might be? Well, we first evaluated their toxicity levels, and we did this in what I would consider the most, well, figuring out toxicity is a rarely humane thing. The traditional ways, you do LD50s, you extract all these skin, you extract their cocktail of toxins, and then you shoot it up into potential vertebrate predators and see what it takes to kill them. I couldn't bring myself to do that, so I did the slightly different version. So we extracted all their skin, and there's a whole breed of strain of sleeping mice. They just like to sleep all this time. So we used this strain of sleeping mice, and we would inject a small aliquot of these toxins into them and see how long it took them to return to sleep, as well as stop doing piloerections and licking. And so only one mouse had to die because we forgot to dilute, or we didn't know how bloody toxic it was for this group. It was an unfortunate situation, but since that happened, we diluted, and that meant watching and watching and watching for really long periods of time. But very few individuals had to die except for our individuals who represented each of these different morphs. But on the top is the toxicity score. It can be either viewed as the time to go back to sleep or stop doing kind of grooming after that injection. And then on the bottom, you have dorsal brightness. So simply taking the reflectance measurements from the dorsum and looking at the total area under the curve. And you can look at these together as a correlation, and it's not bad. There's a simple correlation on the inherent brightness of these different morphs. But as visual scientists, we're more interested in how specific viewers see this relationship. And so we evaluated that the relationship of perceived brightness and color as an overall conspicuousness score. And we evaluated from, well, at that time it was called Dendrobates familio, not Ophaga familio, but we looked at how a conspecific can evaluate the relationship between how toxic these morphs were and their overall conspicuousness, a bird, a crab viewer, and a snake viewer. And if you look across the R-squared values, you might realize or see that the bird relationship is extremely high. It is rare doing environmental physiology work to get an R-squared value of 0.94. So we were ecstatic, and although this is not a smoking gun at all, it certainly is consistent with the idea that that variation in coloration was driven by an avian bird predator. And why do we have variation in toxicity in the first place? It's because these animals actually get their toxins through their diet. And if they're stuck on islands with lower toxic mites and ants, then they're stuck with a less toxic, potent weapon. And so presumably the idea is they had to actually evolve a coloration pattern that actually allowed them more crypsis than conspicuousness. Okay, so I then want to take you on a dive into the ocean to look at how predators shape the evolution of signals or color patterns. And this is the work that Elizabeth mentioned early on. I'm going to get into polarization. So, and this is the part of the talk where I'm going to go from the seen to the unseen, because our eyes don't really pick up on polarization. And this actually is relevant because the first, and I really want to highlight that this work is all done in collaboration with a wonderful biophysicist, Dr. Perch, who has worked with me for a number of years. So it is relevant that we do not see polarized light, because when early different countries wanted to hide things like submarines, they turned to scientists to say, what is the best material and properties of those materials to hide our various things underwater? And in the 70s, people, when they asked this question, they didn't really pay attention to polarized light. And folks in England were asking their scientists, such as Sir Eric Denton, and he said, well, the optics of the homogeneous light field, like the open ocean, which is the same in terms of color and brightness surrounding the fish, you should just use a mirror, because then you reflect back the exact properties on the opposite side, and no one is the wiser. And so this is a fish with a mirror-like property, simulated fish in a mirror-like property in a homogeneous background that's the same in terms of color and brightness. And that worked very well back in those days, and that actually worked very well as a military strategy, because back in those days, we didn't actually have the sensors to detect polarized light. Well, about 25 years later, folks started to actually use polarization sensors, and they could put them on satellites, and they could orbit the Earth, and they realized if they compared sensor types, those that just picked up color difference, those that just picked up brightness difference, and those that picked up polarization difference, that the polarization sensor was twice as sensitive at picking up targets in the ocean than the other two sensors. And that's because you're about to see a whole bunch of reasoning involves why polarization works best in that condition. And I'm going to give a brief primer on polarization before I kind of go into the actual data. So all of us in our room, we're used to evaluating the reflectance properties of various animals in terms of their brightness and color. But a whole lot of animals out there see another form of the light field known as its polarization component. And that's really referring to the vibrational plane of the light waves, and specifically whether or not they're vibrating coherently with each other. And so there are certain places on this Earth where polarized light is more important than others, and they all involve water. So light enters our atmosphere from the sun completely unpolarized or depolarized. It means that those photons are vibrating willy-nilly in every particular direction. As soon as it hits our atmosphere, it interacts with water molecules, and water molecules bias the direction of that vibrational plane, a very predictable direction. So our sky actually has a polarized gradient to it. Same thing happens when the light hits any surface of water. You've got a biased direction of the vibrational plane. We quote unquote see polarized light in these particular situations where we're out in sea and we see it as glare. We're not actually able to pick up angles of polarized light, but we just see a blast of light. And so what we do with our polarized sunglasses is simply block out that specific direction. If you ever want to test whether your sunglasses are polarized, there's a really simple way to do it. If you happen to be out on a sunny day near a water surface, just turn your head 90 degrees and you should get that blast of glare. If you don't, take those glasses back and get a refund because they are not polarized. So as I mentioned before, there are two places on Earth where we have a lot of polarized light. There are places where water molecules are abundant. So up in the sky, we've got a polarized light gradient. And not surprisingly, a whole number of different insects can see polarized light. Some block it out, some try to see it very well. And the other place, that's an interesting phenomenon. I guess we can't see those insects that can see it very well. Am I frozen in time? I just have maybe a connection thing. I have an unhappy computer situation. It's no worries. You can sit back down. And there are a bunch of fish who can also see this. In fact, representatives within 6 different families of fish have been shown to be sensitive to polarized light. So it's pretty, I think it's a safe assumption that a large number of the underwater light organisms can actually detect this amazing field. So why is this relevant to things like the Navy? Well, if our rivals have polarization sensors on their satellites trying to find targets around the world, they could pick things up pretty easily. And why that is because polarization is very angular dependent in terms of not only where the viewer's looking, but where that source of the main light beams are coming from. So when the sun is directly overhead, we get a world exactly the way Sir Eric Denton from London described it. It's very homogeneous around the animal. The main vibrational plane is always orthogonal to the main light rays. So at noon or high noon, when the sun is directly overhead, the main plane will be right around the animal. As soon, and this is a depiction of the degree of polarized light, so it's just how many of those light waves are vibrating together. When the sun goes over and moves in the sky, the main plane, the vibrational plane moves with it, and so it tilts, and yet the fish is not tilting with it. And so on parts of its body or background, it's going to have high degrees of polarized light, and in other parts, in this case, in this cartoon, in front and behind, it's going to have low polarized light. So it's going to be very heterogeneous. This particular problem gets even more pronounced when you take into effect the angle of polarized light. So which particular direction that vibrational plane is vibrating in. And again, at high noon, the Eric Denton world of homogenized light field is present. But any other time when the sun is not directly overhead, where it's maybe close to the horizon, life is particularly complicated. And you can imagine, okay, now an animal has to have, essentially in this scenario, 3 different color or signal attributes it has to be reflecting. One that matches this particular angle on its right side, one that matches this particular angle on its left, and something different in front and behind. And so when we did some simulations for how this might, a mirror strategy, the old world strategy for being a reflector, if you take into account polarization, here the background is the angle of polarized light. And you can see the fish often is clashing. It's reflecting a very different angle of polarized light. So not a good strategy. A mirror would be a pretty poor strategy if your rival had polarization sensors. So this work was actually funded by the Navy. Yeah, shout out to basic research. If you're studying these questions, we all study basic science. And we're one of the few nations in the world that values basic science, and their military looks to basic scientists to find solutions. So I try to make a shout out for basic science kind of being the flywheel for applications down the road. So we began by Perch building this amazing video polarimeter. We then got to go scuba diving with it, and we measured fish and light in various environments. And we soon became enamored with this particular group of fish. Well, we got to go out. I never was a fan of oil rigs until I started doing this project. They become these amazing biomagnets out in the middle of the ocean. So we're at UT. We went out to these oil rigs. My job was to hold on to the oil rig platform and hold on to Perch to make sure he didn't float away while he measured these different fish. And our number one candidate became these Lookdowns, Saline Vomer. It's a group of offshore carangid fish. And they became our candidates because, as you can see here, they look very mirror-like. If you just pay attention to color and brightness, they do an excellent job of blending into that environment. So we brought these animals into the lab, measured them under very specific conditions, and then we did some optimality modeling in the polarization world. So it's a little bit beyond kind of just looking at reflectances. With a Mueller matrix is a 16-term matrix that explains or describes the polarization features reflecting off of a surface. And so we did some optimality where we varied two different terms in terms of their properties and evaluated what would be the best type of reflectance to minimize contrast in an open ocean environment. So what you want to pay attention to here is where you see purple. That's that optimal basin for having a reflectant property that blends in the best with the background. And then we overlaid different measurements of different surfaces. You know, the mirror hypothesis, you know, was way off or definitely off the optimal basin. Some coastal fish had properties that were definitely off the basin. But two different offshore carangid fish had properties that were falling in that optimal spot. Now, I love optimality visual modeling, but what's far more satisfying to me is testing it in the field. So we then convinced the Navy that we needed to test this in open ocean conditions. So we got to go down to Curacao, and Curacao was perfect for this because you could anchor your boat in 20 feet of water, drift out to 200, and be working in a polarized, well-structured polarized world. The polarized world in the near shore is very scattered. The substrate essentially nullifies the polarized light field. So you have to get offshore to have a strong polarized environment. And so here's just an example of the kind of, this was, as I said, work done by the Navy. We measured fish on a platform from all different angles. Well, because I have 5 minutes, we're going to cruise through this. But we put live fish in against a mirror and a diffusing reflector, and then when we'd take these measurements where we would spin all the way around 360 around the fish, and we did it all different times a day. And I'm going to briefly show you what a spin looks like. So this is a video from our video polarimeter. Here's the fish, here's the background, here's the mirror. And this is all depicted in the angle of polarized light. And your idea is you're trying to, across this 360 spin, figure out, does the fish match the background better, or does the mirror match the background better? So we did this across 2 weeks. We took lots of measurements in all different angles. Remember, polarized light is very angular dependent. And after thousands of measurements, we took a simple approach. Okay, who minimizes contrast the best? Fish to background, diffuse mirror to background, or mirror to background. Both of these open ocean species, on average, did better. And they did better than nearshore carangids, okay? Because we're more evolutionary biologists, we actually know that different angles matter in terms of predation. So we looked at predation angles. The approach angle of a predator is 90 degrees off its tail, as well as when this fish is hunting, 90 degrees in front. And when you look at these chase angles, it was an even more significant drop in contrast. And these non-offshore fish species did not show that pattern. So it suggested that natural selection really shaped these reflectances. Real briefly, we did another way of looking at this. Instead of taking all of our measurements at once, we did it from the perspective of individual fish. And we found a similar, we did it across 11, and we saw a significant drop in contrast across all their measurements. And then we just looked at the 5 fish that had the greatest number of measurements, And you can see even across 5, it's a consistent trend. Okay, I'm going to cruise on because I wanted to get to this part. So if we know that fish are natural selection's shaping fish signals to avoid detection in polarized light, then, you know, I just couldn't stop there. I wanted to actually ask whether sexual selection was shaping reflections of fish to be seen in their environment. For here, I went to the freshwater swordtails, used this wonderful device to evaluate signals in males. Here are male swordtails. We know they court females using visual signals. Here's a video polarimeter simultaneously showing the angle of polarized light, the black and white intensity, and the degree of linear polarization. What I want you to notice is a lot of features of these males pop up, and they're reflecting different angles of polarized light against the background. So from a signal detection idea, this would suggest that it's a great prospect for being a potential signal. So we then looked at sexual dimorphism, and we found that males, more so than females, everywhere you see an arrow, these parts of the male body showed significant more contrast in the polarized light field relative to females. We then tested it behaviorally. We set up this is hard to do, to not vary intensity and not vary color and only vary polarization. So we set up this mate choice tank where we illuminated males and females with either a diffusion tank with the polarizer near the male, so this would create a very highly polarized environment, or the polarizer on the opposite side of the diffusion tank, so light would be polarized and then become unpolarized. And so this is the kind of environment we created. We tested it without fish as a control. Do fish like to go to polarized environments or over non-polarized environments? And then with males present. And this is what we found. Females, when no males were present, equally spent time in the polarized and unpolarized environments. However, when males were present, they far preferred to see the males with their flashy bits. And we also, I'm not showing you the third bar in here, but males didn't differentially court whether they were being polarized or not. So there was something about his reflectance that caught the female's eye. Okay, this is the last data slide. It's the least exciting data slide. We should have redone this, but in terms of how it looks, but I think the content of this is pretty exciting. This essentially shows social modulation of the polarization signals. So we can look at how contrasty in degree of linear polarized light these different fish are. And what Gina Calabrese did is we had these tanks with either a fish by themselves or fish with others, males and females. And then we only evaluated, we took male and female photos only when the fish was 90 degrees to the camera, and that's important because everything is angular dependent. And then we evaluated the contrast both within the fish, within body contrast, and against the background. And if you look at the females, whether they were by themselves, which is open, or social, they didn't change their contrast. However, the males, when they were by themselves versus when they were with others, they actually turned on their polarization contrast when they were with others, suggesting the potential for being a dynamic display pattern that could be directed at specific conspecifics. So, okay, this is good timing. I'm ending here, but I just want to point it out. I hope I set the stage for the future game changers who are now going to take us on a journey into the future about how we take this field further. And these are just some of the questions that I think are the direction of the future, not going into unseen, not just going into unusual sensory modalities, but then taking these really strong phylogenetic comparisons, as well as getting to the underlying genetics like Marcus is going to be presenting at the end of the day today. So with that, I will end and thank the funding as well as all the people involved and take questions if there's time.
Hello. Great talk, Molly. That was really stimulating. Just a quick question about your last result that you were showing. If you're keeping the angle that you're taking the measurement consistent, how exactly are the males changing the amount of polarization?
Thank you for asking that. That brings up two really exciting bits. So male fish have a portion of, some fish have motile iridophores. So we think they're doing some dynamic changing with their iridophores. But it also brings up a second point. This is a highly conservative measure because in our fish, they do crazy behavioral displays. So we're not even capturing all that potential contrast that might be there when they're actually doing their full display. So we have a long way to go with this, and this is just that first conservative. Even at this, where we're keeping them there, they're showing a difference.
Great. Thank you.
Mm-hmm.
Just out of curiosity, and looking at a lot of your research has looked at how these, the kind of advantage that many of these color schemes or color signals could have given to the organisms that possess them. Has there been any work done looking at whether there was whether the color signals evolved first or whether a response was given to certain males that could have been eventually paired to a later signal?
That's a great question. That's a great question. That's a wonderful question. Problem is, preference doesn't fossilize, right? So color, you can find some great examples of fossilized color, but you can't, the behavior portion is really hard to capture. So from my vantage point, I think other people have done some good modeling on this. It doesn't make sense that the color would evolve first in these kind of systems because males who have a color and females don't notice don't have any particular advantage until a female can see that. And so once females have a bias, then males who actually have that color have that communication advantage. And so then you can get selection driving and possibly linkage of preference and trait moving forward. But before females can see that, unless that trait, and before females can see that, it's just going to be random. There's no reason it should actually get linked with preference. So in general, although there are some deviations to that rule, but more often than not, it's the receptor has to be there first, or that particular bias, sensory bias has to come before the signal.
Thanks. So, is it working? Yes? Okay.
Yeah.
There were so many interesting things to think about in this talk. Thanks. As someone who doesn't think very much about aposematism, I was kind of surprised that there was that continuous variation. So, like, why isn't it bimodal with some really bright? And, like, what's the point of being mildly aposematic?
Right. So what I think's happening there is that, what I didn't point out in the middle of that continuum is where the mainland morph is, okay? And so the archipelago, the island morphs some are above and some are below. So on some islands, they must have some nasty micro critters there, really nasty toxic things. And so in those islands, the idea is these animals became really, really, really toxic. And you can even advertise more how brightly colored you are and really have the Solarta, they are so bold, they do not move. JP actually helped us it was pretty neat. They are just not going to budge. They are so nasty, no one needs to bother it. But if you're stuck on an island with less nastiness, then actually you want to start moving towards Crypsis because otherwise the local predators are going to learn you're not there. So I agree, it's kind of interesting that there's a continuum of toxicity, and presumably then that what happened next was a continuum of Crypsis to conspicuousness. But yeah. It does set the stage to look at other places besides this archipelago with that question.